So the overall pathway or process for a visual system for the V1 pathway is that you have light hit your retina, and then it's processed through the optic nerve, which is a bundle of ganglion cell axons, and then it goes to the lateral geniculate nucleus, or the LGN. And then the statistic has come up quite a bit. 90% of it goes to there, and that's in the thalamus, and 10% goes to here, which is different. And then from there, it projects over to the visual cortex in your brain, which is the V1 or striate cortex. And then in terms of what goes on in your brain, you have this retinoic map, which is what we usually talk about with the example right here. But I'll get into that more so later. And this is just so that the understanding that you see things in your visual field, and that's like it goes from your visual field, what you're seeing, into your retina, and then from your retina it projects onto your brain. And then this is just knowing that like the central visual field, it's more prominent in your brain as opposed to like peripheral, because you're more focused on it. And then in terms of blind sight, that's just a phenomenon that we've gone over quite a bit. And unlike this whole entire V1 pathway where it's all about consciously seeing something, blind sight is unconsciously seeing something. So what happens in that case is that people are blind, but they swear that they see something, and that's because it undergoes a separate pathway where the output doesn't go through this pathway right here. It instead goes to the superior colliculus. So if anything comes up about blind sight, all you need to know is superior colliculus, and that's a different unconscious pathway. So we also had a few questions about this. This is just understanding binocularity and how that works. So you have two eyes, so you have binocularity as is, but understanding how deep something is, this depth perception, you have this pathway going on, where in this process, the LGN goes to this V1 striate cortex, but in the cortex you have different layers. And so what happens is the LGN directly goes onto layer 4, and then from layer 4 it projects onto layer 3. So it kind of branches off from this like straight pathway to branches into layer 3, and then in this layer 3, that like mixture of different codes from layer 4 that leads to binocularity and depth perception. So that's all you really need to know about layer 3, is that layer 3 is where the binocularity and depth perception occurs. And then quickly going through this again, the three parts you really need to know are the optic nerves right there, the optic chiasm right there, and then you have your optic tracts right here. So remember that vision is contralateral. The only part about it that isn't contralateral are the nasal tracts. So the nasal tracts or nerves are ipsilateral, they go straight. And then you can remember that because in the optic chiasm, if you cut it off, you lose your temporal viewpoint. Temporal being to your side, the outer sides where your temples are. So that's just something helpful, because I feel like that was pretty confusing. If you cut your optic nerves, you still can see most of your visual field. You just lose like this quarter of it, really. Meanwhile, if you cut off your optic tract, you're going to miss out on this whole entire thing. And for that, it's for cutting off your optic tract, it is contralateral. And then for if you cut off your optic nerve, the missing part should be ipsilateral. Okay, so let's talk about the vestibular system really quickly. All you need to know is that it's necessary for balance. This is the video we watched about like people carrying stuff and then falling over whenever they saw the slightest movement that messed with them. And for this, you can basically read the slides pretty straightforward about how that happens. There's a vestibular ocular reflex, and that explains it quite well. So beyond V1, you have another two pathways. There's a dorsal stream and the ventral stream. And then with dorsal stream, that's looking at area MT, middle temporal. And for this, it's just knowing that you have clusters of neurons in your brain that turn on when you see things move in a specific direction. And then you can manipulate it where if you turn on those neurons, you can get an animal to believe that things are moving in a certain direction, even if that's not the direction they're actually moving in. And then you have the ventral stream, which is where we talked about the different diseases or impairments. And so there are two areas to ventral stream. There's area V4 and then there's area IT. So for V4, that's color and shape. So you have achromatopsia, which is the color blindness. And then for area IT, you have face recognition. So you have prosopagnosia where you no longer recognize someone's face, but you can tell that to some extent there's recognition via the skin conductance response test. Not to be confused with SCN, as I so previously made that mistake earlier on. And then you have the second one, which is Capgras delusion. If you want to learn more about SCN, I wrote it right here so you can understand the process behind SCN. With Capgras delusion, you are unable to do SCR. Sorry, I totally said SCN again. SCR. But you're unable to do that because there's impairment beyond area IT, most likely in your amygdala, which ties into the last few lectures that I don't think I will be getting to. So next we have the auditory system. With sound, there are two things about it. It is frequency and amplitude, and sound is air vibrations. So those were the two things that really sum up what sound is and how we perceive sound. She talked a bit about visualization and quantification of sound. So if you look at an audio waveform, you can see it in 2D. If you look at a spectrogram, you can see it in 3D. I don't think there's anything really important to know about that. So in terms of the process, there are three processes involved. You have air vibration, which is when the sound enters your ear. So it goes from your pinna to your auditory canal to your eardrum, which is otherwise known as the tympanic membrane, which brings you to the next thing, which is ossical vibration. So ossical or mechanical vibration, it's when the three components of the ossicles are moved and they act as like amplifying levers. So the three are the malleus, incus, and stapes are connected to each other like little joints, like 1, 2, and 3. And then this vibration pushes them, which then amplifies and pushes us on to the next step. So the last step here is the fluid vibration. So these ossicles push against the oval window, and when they push there, it causes the fluid inside the cochlea to move. And then in the fluid are hair cells, and these hair cells are the receptor cells in this case. And then that's in the organ of Corti. You don't really need to know anything specific to that. Moving on from that, what happens is these hair cells, because they're moved, because the fluid moves them, they synapse onto the spiral ganglion cells. And then the spiral ganglion cells send information into your brain through the auditory nerve. So this is what happens in your brain. This is, again, where it ties into what we're so familiar with action potentials and processing. So specifically hair cells, because that's the receptor cells, they have something called stereocilia on them, which are like these little things. And then on the stereocilia, you have channels. And what's happening in this case is that you have this liquid called the endolymph. And outside in the liquid, you have a high potassium concentration. And then on the inside, you have low potassium concentration. So when the channels open because of the movement, you have an influx of potassium. So again, if you have an influx of potassium, you have depolarization because it is now becoming less, well, actually in this case, it's becoming less negative. I know that's kind of confusing, not to be confused with sodium and what we usually learn, but think about it in terms of high to low. If it's going from high to low, it is depolarizing. If it's going from low to high, then it is, or it doesn't go from low to high, but if it's creating less of a gradient, it is depolarization. Depolarizing is what I'm trying to say. And in the case of depolarization, you have your calcium channels that open, and then vesicle fusion, and then neurotransmitter release. And in this case, it releases onto the spiral ganglion cell.